Okay, good morning, guys. Can you hear me? So, I think uh, we have to start our tutorial. Okay, in this case, do you remember our first three lectures, which I have uploaded? I mean, my online lectures. So, we discussed the embryology, general, different topics of general embryology. But now today, I want to talk with you about the different samples, different type of microscopical techniques, and describe together with you and study together with you different tissue samples via the virtual microscopy. So usually, when we have usual lifestyle, we are working at the e-classes at the university where we have light microscopes, tissue samples, and we have possibility to see with our own eyes what is happened in light microscopy. Hopefully, you have seen light microscopy and you have even small experience to work with light microscopy. So, using virtual microscopy, we can observe not only light microscopical uh, views, micrographs of tissue samples, different type of cells, but also we can observe their quite very fine and detailed structure using the transmission electron microscopy and also three-dimensional images of scanning electron microscopy. So let me to start. Hopefully you have, or if you do not know, you will see it now and you can use this web page in the future. We have several web pages which are written in your syllabus using which we can observe this tissue samples. So look, very important when we are talking about the different type of cells. Very important parameters are size of cells, for example, or shape of cells, or nuclear cytoplasmic ratio of cells. Do you know what does it mean, nuclear cytoplasmic ratio of cells? It means ratio of their sizes to each other, because we have some type of cells which have quite big nucleus and very thin rim of cytoplasm, or very small nucleus and very big rim of cytoplasm. Also very important is um, chromatin, level of chromatin condensation, or which part of chromatin is in prevalence in the nucleus, for example, heterochromatin or euchromatin, because it gives us possibility to make a conclusion or um, consider the level of activity of the cell. Also, very important is staining color of the and type of the cell. For example, cells might be stained basophilically or they might be stained acidophilically. It depends what type of stains are first of all used there, and also very important is which part of cell is well stained. I mean, nucleus or cytoplasm. We can also identify in the cell's granules, secretory granules, using which we can also, of course, study, make a decision about the type of cells. And also some cells, have special staining properties to see them clearly, to identify specific type of features. Very important is this special type of staining. We For educational purposes, usually mostly are used light microscopic images, TEM images, I mean transmission electron microscopic images, and SEM images. It means scanning electron microscopic images. You know, we have what we can do, for example, let me make a brief description how briefly describe how the light microscopy is working. So it has source of lamp, which is a source of light, which is lamp actually, and then this beam of light move through the system of lenses, which is called condenser lenses, and usually it is allocated beneath the stage. So then is the stage itself where the usually tissue sections are allocated. So the condenser lenses are able to focus this beam of light on the tissue sample. Then we have objective lenses, and objective lenses from send the light to they magnify first of all the image, and then they send this image to the eyepieces, which also are set of lenses which magnify additionally the image, and finally our eyes are able to um, identify and to see the uh, field of vision. So um, this is the main, how to say, very brief, very superficial description of the light microscope. What has happened in the case of transmission electron microscope? There, the key player is beam of electrons. So the tissue samples, of course, should be processed appropriately and differently for light microscopic observation for transmission electron microscopic observations. So what is the most important thing there, that uh, tissue samples should be very thin because the beam of light should pass through the tissue sample. Only in this case we can see what has happened, how the structural arrangement is, and how everything is arranged in the cell or in the tissue. That is why this is really very important. So the when we are working with the when we are preparing tissue samples, first of all, as you know, we have to fix these tissue samples, and immediately when we fix these tissue samples, what is happened with the cells and with the different metabolic pathways in these small tissue samples. Of course, everything is stopped there. The main goal of fixation is to preserve internal structure of the tissue, internal structure of the cells. So, and here the key players are different type of fixatives, which are different in the case of light microscopy, in the case of transmission electron microscopy. So after the steps, the following steps, I mean, Then we have, for example, we have to use, besides the fixation, very important is dehydratation. And using the dehydratation, we remove from whole, tissue sample, um, the fluids, any kinds of fluids. Uh, then very important is to put this tissue sample in the uh, clear. Next step is clearance. When we put, for example, this tissue sample in toluene, these are different. This is a little bit another type of fluid which is able to remove the ethanol which is used during the hydration from the tissue sample. After the clearance, we can put this tissue sample into the melted uh, artificial um, substance which is used for embedding. For example, in the case of light microscopy, usually is used paraffin or any types of other artificial resins. So these resins or this paraffin, melted paraffin, at quite a high temperature, for example, 57 or even 60 Celsius, um, the infiltration process takes place very actively. It means that the melted artificial embedding material um, in, infiltrates the tissue sample. And then we have to put this tissue sample into the um, again melted resin or paraffin, and uh, we prepare finally blocks. So what is this block of tissue? This is small amount of tissue sample which is embedded in the block of paraffin or block of uh, epoxy, for example, in the case of electron microscopy. And then we became able to section this tissue sample. And as a main result of sectioning, we receive very thin tissue slices. So you can use the keywords, for example, tissue sectioning or preparation of sections for light microscopy or transmission electron microscopy, and see this everything online in the online regime. So a lot of different type of videos are available in the internet. Hopefully when everything will be finished, I mean this pandemic situation in Georgia and in your countries and everywhere on our globe, so we will have possibility to work with and to see real processes, the real these processes in my lab where I am working, and also in the lab of New Vision University. So the New Vision University will have very soon, uh, very, how to say, very advanced and cutting edge lab in the university. So we will have the confocal microscopy, I'm not talking about the light microscopy. Of course, light microscopy will be there too, but most important is, I think, this cutting edge microscopical techniques, which is the electron microscopy, confocal microscopy, etc. So, okay, let's back to our tissue samples. So, as I have told you, we prepare this thin uh, tissue slices, and then we put these tissue slices on the glass slides. Glass slides is covered with appropriate substance, and via this substances, the tissue slices are adhered to the surface of glass slide. And when this is everything ready, and when it is um, uh, undergoes drying, so we can stain this tissue sample. Without staining, the details of tissue sample is not so easy observable, of course. Uh, we have unstained tissue samples, for example, in the case of live tissue samples. I want to show you the image, sorry, which I use for my lectures. You can see this in your presentation. So, mm, just a minute. Okay, methods. And look what you can see here. Do you remember this image? We see here unstained, not stained tissue sample. This is light microscopy of living epithelial cells, and they are unstained. But at the same time, you can see they are alive, and they are prepared via the method of tissue culture or cell culture. So what do you know what does it mean, tissue or cell culture? It means that we maintain and observe the live cells in artificial conditions. So this, for example, the same epithelial tissue which is given here, this one is fixed, this one is stained, and this one is sectioned. This one is not sectioned. Okay, so we provide the growing of the cells, maintain their viability in the petri dishes, and here this, and we observe them in artificial medium, etc. So but here the cells are first of all fixed, and then um, they might be also sectioned, but these cells are not sectioned, they are fixed and stained. Okay, but this, for example, this tissue sample is tissue sample prepared via the transmission electron microscopy. This is fixed, this is sectioned, I mean tissue sample, epithelial tissue sample, and also it is stained. You see that these cells are stained too. Here I want to talk with you about the staining. This is quite important moment, you know. Uh, Look, we have two main types of um, dyes which are used in histology for educational purposes. They are basic dyes and acidic dyes. So the main difference between these two type of dyes is charge. Basic dyes have a positive charge, they are positively charged, and they usually bind to components of cells that are, of course, negatively charged. And such type of components in the cell, in the tissues, are nucleic acids, DNA, RNA, and also sulfated polysaccharides. So these sulfated polysaccharides, um, glycosaminoglycans, you can identify in extracellular matrix, in the cells too, but at the same time, well stained is mucose. Mucose is um, a type of substance which is synthesized by the cells, released, and when they release this substance, it is mucin, and when it is highly hydrophilic. Hopefully, you know what does it mean, hydrophilic substance, and they attract a lot of water, and they became um, mucose. So mucose. Another type of staining which we have is basic dyes. So the basic dyes are very important for to demonstrate, for example, the proteins in the um, cell, tissue, etc. So because this acidic dye is negatively charged, and this, of course, it is attracted and easy bind to the everything what is charged positively, and such type of charges we have in the proteins as a amino acids. So in the case of DNA and RNA, in the case of basophilic staining, you see here phosphate groups which are charged negatively and which are able to attract the and bind the basic dye. That is why very well stained will be everything in the cell what contains nucleic acids. So what type of structures in cell contain a lot of nucleic acids? Of course, nucleus, first of all, and also we can identify cytoplasm too. So in the cytoplasm, we have rough endoplasmic reticulum, which contains has ribosomes on the surfaces, and ribosomes, as you know, contains ribosomal RNA plus transfer RNA is also represented in the rough endoplasmic reticulum. So that is why all these structures are very well stained. Um, Also, as you know, the DNA might be identifiable in the um, mitochondria DNA. So, but mitochondria is not actually well visible in the light microscopy. It needs special type of staining to see where location of mitochondria. But rough endoplasmic reticulum uh, might be seen as a some type of granulation in the cell cytoplasm, and I will show you this. Another one is acidophilic staining and acidic dye when we, for example, um, which stains very well any kinds of proteins. And proteins, as you know, you can identify almost in all types of cells, and almost everything in the cell, in the tissue, contains this substance, proteins. Okay, so now I want to show you this different type of stainings and tissue samples which are stained with this two main type of dyes, basic dyes and acidic dyes. You see here that using this web page, I mean histology guide, we can see not only tissue samples, but we can using different type of methods. I mean microscopical method, but we are also able to change the magnification and see the details, fine details of the cell. Okay, let's go back and observe the tissue samples. Tissue samples which are stained using the basophilic staining and using the acidophilic staining. Look, what is here? What we see here is tissue sample from the pancreas. And this is type of dye which is called hematoxylin. This is basic dye, and using this basic dye is stained this tissue sample. So we can change here the magnification, and using the magnification we can see fine details of this tissue. But this is quite small magnification. You can see quite big area of quite big part of tissue sample. Um, you see here the connective tissue, mm, also together with the epithelial tissue here. So this is stromal part of this organ. This is parenchymal part. Um, these are the blood vessels, and usually the uh, tissues are divided in two main parts. This is stromal part, and this is parenchymal part. So the stromal part contains mostly connective tissue, which is very well vascularized, and you see this white not stained part is stromal part, and it contains the blood vessels, and plus you can see that this tissue itself, the sample of the tissue is low. Um, Besides this dark stained portion, these dark stained lobes of the tissue, they are a parenchymal part of this organ. So the parenchymal part usually contains this specific type of cells which accomplish the function of the organ. So if we magnify this image, for example, look, step by step we can magnify it. Look, so this is stromal part. This is blood vessel, arteriole, for example, venal also. Here is again arteriole. Look, arterioles are mainly round shaped, venals not. And looks the same as here. For example, arteriole, venal. And what has happened, how the tissue, how the parenchyma looks like here. Look, okay. So what you see here are islands. Look, of cells. Here the cells are allocated in the groups, and you see here, for example, the cell itself, another cell, another cell, another one, another one. And first, when we look at the tissue sample, what is very important, you have to be able to identify the nucleus. When you see the nucleus in the tissue sample, much more easier is to identify the whole cell. So look, we see here nucleus, we see here nucleolus, and then we see the something what surrounds this nucleus. So this something is the cytoplasm. And you see here the borders of the cell. Look, I am use this word borders because using the light microscopy we are not able to see the cell membrane, because cell membrane is very thin. very approximately 8 from 8 up to 10 nanometers. Do you know nanometers, this unit of length? So a centimeter, as you know, has contains 10 millimeters. So this each millimeter contains 1,000 microns, and 1 micron contains 1,000 is equal to 1,000 nanometers. So this is a really very small unit of the lens, but not smallest, because we have also, for example, angstrom, which is used in transmission in electron microscopy, and 1 nanometer is equal to 10 angstroms. So back to cell, we see cell, we see quite well the nucleus. Nucleolus is also visible there. You can identify dark stained part of the nucleus. Look, these blocks of dark stained something, and this something is chromatin, of course, heterochromatin, and a much more light stained part is euchromatin. So when we have in the cell prevalence in the nucleus, prevalence of euchromatin, I mean light stained portion, light stained content of the nucleus, it means that this cell is very active from point of view of gene expression, and it means that the also translation process and cell synthesis is sorry, protein synthesis is also very intensive in the cell. And because these are secretory cells, and because you see a lot of um, dots in the cytoplasm. So, first of all, uh, these are uh, these might be the uh, very well developed rough endoplasmic reticulum. Look, this is much more dark stained portion. And here are the secretory granules and secrets which are synthesized by these cells and then released outside. Okay, so this is small magnification. Look, when we use this small magnification, in this case, we can see quite big part of tissue. So quite big, the field of vision is quite big. But when we, for example, um, magnify image, the field of vision became smaller and smaller. So the maximum of magnification here is 40. Okay, so blue stained tissue sample, mostly blue stained, and here is used the basic dye, which is called hematoxylin. That is why we can see very well stained nucleus, borders of the cell, which actually contains cell membrane and macromolecules attached to the outer surface of the cell. That is why we can see here some type of granulation in the cytoplasm, and this granulation contains, of course, the rough endoplasmic reticulum too. You see clearly in the nucleus itself, nucleolus, look this dark centrally or peripherally allocated uh, quite big dot, and plus we can identify here using this type of staining, we can identify here euchromatin and heterochromatin, and make a conclusion what type of level of activity this cell has. Okay, So, what type of staining was this one? It was basophilic staining. Let's go to another type of staining, which is acidophilic. And for acidophilic staining, we use usually iodine. This is acidic dye, and you can see that it is able to stain tissue samples, usually, color from pink to quite dark red staining. So here is stained everything what contains the protein. So if we magnify the image, look, of course, again, you can see here cells, for example, look, a lot of cells. First of all, you have to identify nucleus, as I have already told you, and then the whole cell. Look, a lot of cells, which are stained pink, more dark pink, then even red stained. Look, venal, small arteriole, venal is here too. So the again the stromal part is not stained or lightly stained. Blood vessels, of course, they are stained very well. Stromal part usually contains the connective tissue. Look, oh, what? Okay, so you see here adipose tissue. Also, the collagen fibers are identifiable, but the parenchymal part is constituted by the cells which accomplish the specific function of this organ. Look, you can see here capillary in the red blood cells in the capilla, in the lumen of the capilla. So you see that everything here is stained from uh, pink to red. And if we compare this type of staining with a basophilic staining, uh, I think basophilic staining is more informative because you can clearly see their nucleus. Look, nucleus, tissue, cell itself, even nucleolus. In the case of acidophilic staining, when we use eosine, they, um, we can't identify so well and so clearly. But however, it is of course possible. You can see here, for example, the nucleus, nucleolus, but identification of, for example, different type of chromatin, hetero and euchromatin, is not so uh, easy. But if we use, for example, these two types of stainings, basophilic and eosinophilic, look, I think picture is much more interesting and much more informative. What I mean, you will see after I magnify the image. I look here. You can see. Sorry. So here you can see the cells with a nucleus, with a nucleolus, cytoplasm of cells, which is part of cytoplasm is dark stained, dark blue stained. Part of them is more reddish look, and uh, nucleolus, heterochromatin, euchromatin, granulation in the cytoplasm. This everything is clearly visible. So this co-staining is very effective tool to identify cells in the um, samples. Here you see another part of the tissue which contains duct, lumen of the duct. Okay, I will click directly this image and you will see. Look, so epithelial cells which line the wall of the duct, lumen of the duct, very well visible nucleus, nucleolus also is clearly identifiable, chromatin, hetero and euchromatin. And plus you can see here also um, cell borders very well. Look, we have here profiles of cells which have no nucleus there. Look, I want to show you this. For example, this one here. Look, this one or that one. Small, some parts of nucleus is only visible. Why it happens? Why this or that cell, profiles of this or that cell has no nucleus? How do you think? Of course, due to the plane of section. In these plane of sections, the nucleus are included. Here not. They might be above or below the plane of section. Okay, hopefully with staining, I mean basic dye, acidic dye, basophilic staining, acidophilic staining, or co-staining, everything is clear. Let me to summarize this topics. So, uh, first of all, we have different type of dyes, and using uh, these dyes, we are able to stain different parts of cells well. And these are basic dyes and acidic dyes. Method itself of staining might be basophilic or acidophilic. And when we use this different type of stainings, different parts of cells um, became well stained. So the basic dye stains very well everything what contains nucleic acids and sulfate groups. So, and that is why nucleus is well stained, nucleolus, chromatin is visible in the stained tissue samples. You can even identify, for example, the acidophilic, oh, sorry, the euchromatin and heterochromatin. You can clearly see borders of the cells, mm, which is also very well visible in acidophilic staining. So we can see also the, what else? Rough endoplasmic reticulum, location of rough endoplasmic reticulum, not, of course, fine details of these cellular organelles. So the fine details of cellular organelles you can identify very well using the transmission electron microscopy. Now we will use this type of microscopical technique. So let me go to another part of this web page. This is electron microscopy. So look, so first of all, here are described these methods via which we can obtain main principles of these microscopical techniques. So in the case of transmission electron microscopy, so what was happened in the case of light microscopy? The light source, which is lamp here, gives rise to the beam of light, and the beam of light moves through the condenser lenses and then to the stage where the tissue section is allocated. Then the condenser uh, focus the beam of light on this tissue sample. Then we have objective lenses, then eyepiece lens, and finally we obtain the image. In the case of transmission electron microscopy and scanning electron microscopy, we have again, of course, condensers which focus the beam of electrons. So the beam of electrons are obtained from electron source, and it is focused on the tissue sample. And from the tissue sample, it is it goes to the projection lens, and from projection lens to the eyepieces, and we can finally obtain the image of the again, the same situation is here. The beam of electrons should pass through the tissue sample. What is not happened in the case of scanning electron microscopy? They do not pass through the tissue sample. They are reflected from the surface. That is why we can see here surface image, but we see three-dimensional image. So in the case of light microscopy and in the case of transmission electron microscopy, we see here the two-dimensional images, and in the case of scanning electron microscopy, we see here three-dimensional images. Okay, if we go to the images themselves, so this is, okay, diagrammatic, schematic image of the cell, and you see here the different parts of the cell. This is epithelial cells. Look, epithelial cell, three-dimensional view, and you see here epithelial cell with the free apical surface, and on the apical surface you can identify these small protrusions which are called microvilli. Uh, each of them is microvillus, and the in the plural microvilli. So They are attached to the surface terminal web, which is represented with a different type of different components of cell cytoskeleton. And beneath this apical surface, we can identify here a whole cell with a nucleus and with a rough endoplasmic reticulum which surround this nucleus. You can see here, look, Golgi complex, schematic image of Golgi complex with a C phase and T phase, trans phase. You see here also mitochondria. You can see also here centrioles, endoplasmic reticulum, rough and smooth. So, and you'll see that the cell itself is located on the basement membrane. Look, this is basement membrane, which contains different parts of extracellular matrix, and this extracellular matrix is produced by these cells themselves. These and other type of cells, I mean connective tissue cells, which usually are allocated beneath the epithelial covering. So you see that yeah, the cells have here um, different some type of polarity, structural polarity. I'm not talking about the electrical charge. No, I'm talking about the structural polarity. And what does it mean, this structural polarity? It means that different parts of cells contain different structural structures, or it is structurally uh, arranged in different way. And this is, of course, determines this function also. So here I received the question, will be this lecture uploaded on the portal or not? You know, this is actually not lecture. This is tutorial. And you can use the this web page, I mean, histology guide, and it will be very good if you will search this web page and see this different type of images. But this is of course not lecture. Lecture regarding the methods, I think, is already uploaded on the um, drive. I will check, and if not, I will upload this lecture. Okay, so this is schematic image of epithelial cell. Epithelial cell, which is arranged on the surface of basement membrane. Part of basement membrane is produced by this cell itself, and beneath is allocated the another part of basement membrane, which is produced by the fibroblast cells of connective tissue. You see that the epithelial tissue is not well vascularized. That is why it is always followed by the um, connective tissue, and via the, which is always quite well vascularized. And using this connective tissue, epithelial cells are able to obtain nutrients and release their waste products into the interstitial fluid first of all, and from interstitial fluid these components move to the cells or to the blood, from blood to cells, and from cells to blood. Okay, let's go to the, if you need, I can give you 5 minutes break. Okay, 5 minutes break, please. If you will, if something is not clear, please feel free and write the questions, or Okay, I will check here your what is written here. Sorry, can you speak a little loudly? Something, a voice really low. Okay, I will try. Maybe something is not well with my microphone. What what about what now? Is it better or not? So sorry, we can really hard what you are saying. Could you please speak up? I don't hear half. Is this lecture will upload it on the portal? Okay, so what about the hearing? Can you hear my voice? Please write me, okay?
Yes, we can hear your voice.
Thank you, thank you. Now it is better, yes?
Yes,
So, was everything clear? Was everything understandable during my speech? What about the
yes,
staining? Do you understand everything? If not, please write me or ask, okay? So I am
okay.
seeing, I see here your comments, so please feel free and write what you want to, for example, re-explain or repeat during my in my speech. So no, because we could not hear you before. Oh, look, how do how to identify nuclei? Okay, how to identify nuclei? I will explain it again. How to identify nuclei? Okay. What else?
Is it because the dark color?
Dark color. Okay, I will go back to the. Oh, you have 3 minutes of break. Do you need it, or I have to study again, prolong my lecture?
We don't need it. Don't know.
Okay, 3 minutes are left. It will be much better if I see your faces, guys. So, mm, for example, Anisa Salim. Why I can't see your faces? Hi, hi, Mohammed. I see you.
Hello. Yes, I can
Hello,
see. Hello.
hello, hello. Nice
Oh,
to
oh,
see you.
same here.
So, what about other students? I can't see them.
Everybody has switched off their video and microphone, so they can
Okay,
hear you.
okay, 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 okay. I think
We are not in good shape. We're not ready.
Yeah, that's what I said. I'm not in a good shape.
yes,
Sorry, I'll be there whole time.
I have seen your message. So yeah, yes. So really unusual lifestyle, really absolutely new reality. I don't know how we can.
Next time I promise I'll do that.
Okay, 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 okay. So, guys, I go back to my explanations. So, here was the question how we can identify the nucleus in the tissue sample. Okay. So, what is this image? Okay, I go to the, so this is schematic image of the cell. Let's go back to the tissue sample. First of all, I will go to the light microscopy images, okay? For example, here, and this is small magnification. Of course, using small magnification, it is impossible to identify cells. Very important is, beside the magnification, as you know, uh, I have mentioned this parameter of microscopical technique during my first lecture. Very important is resolution power, as you know. So resolution power is really the very powerful tool using which we are able to identify the finest details of the cell, even which are, for example, have sizes of several nanometers. So if you, when we magnify, to identify cells, we have to magnify the image. Okay, light microscopy resolution power is quite high because we can see here cells. Look, so conglomerates of cells, group of cells. How we can identify nucleus? We have to remember that nucleus is usually round or um, uh, ovally shaped. When you want to identify cell in the tissue, you have to know its characteristics. I mean, main morphological features. For example, cells of epithelial cells, and here we have epithelial cells. They, or for example, what are the main features of epithelial cells that especially secretory cells, they have quite round, mostly round shaped nucleus, or it might be ovally shaped, and quite big amount of cytoplasm which surround this cell. And when we know that the appropriate staining is used, for example, basophilic staining, yes, or co-staining. You know, eosinophilic staining is not usually used together with a basophilic staining. So when we know the shape of the nucleus, and when we know what type of staining is used. So, first of all, round shaped structures or uh, ovally shaped structures here are given. Look, this one, that one, this one. And using basophilic staining or co-staining, yes, I mean, basophilic and acidophilic staining, you can see this round or ovally quite dark stained profiles. So these are nucleus, okay? Or, you know, nucleus might be stained so dark that the nucleolus is not clearly identifiable. This is usually happened with the nucleus which are less active. I mean, from point of view of gene expression, you know that whole time some set of genes is expressed in the cell because they actually determine functional activity of the cell. And of course, appropriate proteins are translated in the cytoplasm. So you see here this nucleus and cytoplasm which surround. I will give you another tissue, show you another tissue samples, for example, where we have to be able to identify this cell nucleus and cells themselves. Look. Nuclear morphology, how to identify, how to see, and very important is also the experience. As much as many uh, tissue samples and micrographs you have, you will see, uh, much more easier you will be able to, much more easier will be for you to identify different parts, components of the cell and cells themselves. So look here, this is lymph node. This is part of spinal cord. So lymph node and spinal cord. What is here? Liver. This is part of, small part of liver tissue. Look, I think first what you see here are nucleus, because nucleus are usually the more dark stained than the cytoplasm of the cells. Here we have a longitudinal section through the blood vessel. Look, and you see here RBC and also leukocytes. Look, this dark stained, they are, I mean, dark stained nucleus, they are leukocytes. RBCs have no nucleus. That is why they are stained pink to red. But these cells in the blood capillary, blood vessel, look, this is, you can see the dark stained nucleus of leukocytes. This is tissue. Look, capillary. Capillary is here, and in this capillary you can see only one RBC. Usually the diameter of capillary fit the size of the RBC, because RBC should move through this lumen of capillary. Of course, here this is lumen of the capillary. Okay, hopefully you see here the nucleus very well. These are epithelial cells, and these are nucleus with a nucleoluses. Okay, plus you can see in this nucleus quite densely stained, dark stained um, part, which is in the nucleolus itself and on the periphery of the nucleus. This is heterochromatic. But in the middle, you see the much more light stained part of nucleus, which is euchromatic. Okay, so let's go to this is tissue, liver tissue, okay? Cells, epithelial cells, a lot of cells. Again, blood vessel. Blood capillaries, look, profile of blood capillary. And these are RBC. Let's go to this is parenchyma of liver. Let's go to the lymph node. This is quite big magnification. If we minimize it, we can see the whole structure of this organ. You see here, for example, the different types of tissues. Connective tissue capsule, which surround this lymph node. And if we magnify it, look, we see here connective tissue. This is white adipose tissue. These are cells too. Look, these polygonally shaped structures, which are not stained. These are cells, white adipocytes. This is nucleus of white adipocytes. Look, this dark stained. And what you see here, and small amount of cytoplasm which surround the nucleus and which surround this whole cell together with the cell membrane. You know, when you will study adipose tissue, you will know that the adipocytes are filled with a droplet of fat, one big droplet of fat. And that whole amount of cytoplasm and nucleus is dislocated to the periphery. So this is cell, this is here membrane, should be here membrane, and should be small amount of free of fat cytoplasm. All other part of cytoplasm is filled with the fat. And you will also know that the nucleus is dislocated to the periphery. So you will search the peripheral part of the cell and try to identify their nucleus. Look, what the characteristic feature of this nucleus is that it is quite densely stained. You can't see here the nucleolus. And we have here the prevalence of heterochromatin and less amount of euchromatin. It means that the cell is less active. We can't say that it is inactive, no, but less active than, for example, this nucleus or this cell. Look, or for example, this one here or that one here, because we can see here the small granulations or small blocks of dark stained material, which is heterochromatin. And also we can see light stained blocks of the nucleus, which is much more light stained, and this is euchromatin. So what is the main difference between the heterochromatin and euchromatin? The main difference is that in the case of heterochromatin, DNA is very tightly packed, and that is why it stains so dark, so uniformly by the hematoxylin. This is very good sign, very good indication about the transcriptional activity of the DNA. In the case of, it means that the DNA is, the gene expression is not so active here, because when the DNA is tightly packed, it is not so well reachable for different type of transcription factors and substances which are needed for transcription process. In the case of euchromatic nucleus, I mean nucleus where the euchromatin is mostly represented. In this case, of course, DNA is not so densely packed, and in the case of euchromatic nucleus, we can see these small blocks of or clumps of chromatin, which is stained dark. But the prevalence, the most part of the nucleus is light stained. For example, like here or there. So you can see this dark stained parts of the nucleus only on the periphery. Okay, why I have started to talk with you about this to identify how we can identify nucleus. Look, this is parenchyme of the node, lymph node. So what we see here different type of cells, and nucleus are very well visible. Look, these dark stained dots, all of them, they are nucleus. Look, dark stained nucleus, which are surrounded with a thin rim of cytoplasm. Lymphocytes, look, lymphocytes. Plus we can see here also quite big cells, look, with a quite light stained nucleus. In these places, we are able to identify easier, for example, macrophages, connective tissue cells, or we can see here also fibroblasts. Look, fibroblasts. Or red blood cells without nucleus. Blood capillaries. Okay, so first of all, what you see in this tissue sample is nucleus, okay? So this is a lymph node. Let's go to the spinal cord. Look, this is spinal cord. And in the spinal cord, of course, we can see cells of nervous tissue, and they are neurons. I will show you neurons of different size. The most important, I think the most, not most important, but I think the most interesting part of human body is, of course, the nervous system, especially brain. Uh, because maybe because I work at the brain ultrastructure and nano architecture lab. So this is the title of lab where I'm working as a researcher, and I think this is amazing organ of human body. So look, you see here neurons. This is neuron. This one too, for example. This is big one, this is small one. Also, you can see here small nucleus, which belongs to another type of cells of nervous tissue, glial cells. But what I want to show you is the big neuron. Look, huge neuron, very big nerve cell. This is nucleus, and this one is cytoplasm. Here is glial cells with a quite dark stained nucleus. Another neuron. Look, another neuron with a nucleus and cytoplasm. So quite well visible nucleus is here. Blood capillary again. So look, beautiful, huge, very big cell, neuron. So this is a bigger one, and here you can see. Look, this thread-like structures. So these are structures which actually are derived um, from the cells themselves, because neurons have a lot of processes, axons, dendrites, and via these axons, dendrites, they are intensively interconnected in the nerve tissue. Another nucleus and cytoplasm here. Nucleus and cytoplasm there. Look, blood vessel, look, and a lot of red blood cells. Again, processes, neural processes, and neuron, huge neuron, another one. So usually they are. Have you heard about the gray matter and white matter of spinal cord and brain? So look, this is gray matter, which comprise the cell body and the cell bodies and also their processes, of course. And this is white matter. White matter, which contains mostly processes of the cells. And these processes, axons especially, they are myelinated. So again, gray matter with the cell bodies. Okay, in any case, you see that the nucleus is the most dark stained um, part. And when we are able to identify nucleus, then first what we have to do when we look at the tissue sample and we have to identify type of cell, we have to know its morphological features, size approximately, shape, how they are shaped, and what shape of nucleus they have, what is the cell usually active or less active. Everything this will help you to identify the nucleus and then the whole cell. Okay. Much more easier is to see this everything uh, in the electron microscopy. Maybe you have question. Please feel free, write me the questions, okay? I will check, maybe someone of you has. So we can't really heard what you are saying. Could you please speak? Oh, okay. Lecture will upload. So I have seen this everything already. Now it's a little bit better. So still not a good. How to identify nuclei? Okay, I try to answer your questions. And if hematoxylin using stain allows a better view of the cellular structure, in what context would someone need to use a basic or acidic stain alone? Okay, good question. I will explain. Sorry, not in good shape. Okay, Mariam. Um, you know, so the question is if hematoxylin using stain allows a better view of the cellular structure, In what context would someone need to use a basic or acidic stain alone? Mostly basic staining is used to, I will show you the example and then explain this, okay? So, where is my, mhm. I will go back to, and then to the image. I will try to answer this question and then we will go look. So, brain, part of brain, for example, cerebellum. Look, there are some types of cells which are very well visible using the basophilic staining. For example, neurons. And they contain a lot of, look, for example, when we are working with the brain tissue, uh, we use we can use basophilic staining, toluene in blue, and identify the different types of neurons. For example, here you see two types of neurons. This is Purkinje cells. Look, this one. And these one are granular cells. Both of them are neurons. But when we have, for example, possibility to stain the neurons and to identify how structurally the cells are arranged in the this or that brain structures, we usually use this uh, staining, this type of, I mean, basophilic staining, because cells are very well visible. Nucleus and cytoplasm is very well stained. You know why? Because the neurons have a well-developed cytoplasm with the rough endoplasmic reticulum. So look, cytoplasm became very well stained, and the shape of the nucleus is clearly, a shape of the whole cell is clearly visible. And of course, size too. That is why we became able to identify about what type of cells, what type of neurons we are talking, how they, what type of, what shapes they have, what size they have. When we use, for example, the tissue samples, and when we see here cells, we can even measure the cells, you know, because always the cells, the scale bar is always represented on the image. And via this scale bar, we are able to measure the cells. So mostly this different type of stainings alone, and especially this basophilic staining, is using to identify subtypes of cells, their shapes, their morphology of the cell. Okay, so you see here the Purkinje neuron, and you see granular cells. These are also neurons. Look, but they are not. They have totally different size. They have totally different shape. Or, or this one, for example, or that one. Granular cells have so small rim of cytoplasm that it is not even identifiable sometimes. Okay, here you can see the small rim, or here, look. But you know, this type of staining, I mean acidophilic and basophilic staining, they have some minuses, of course. For example, in the case of nerve cells, or in the case of connective tissue, when we want, for example, to see whole shape of neuron, uh, we have to use the silver staining. Using the silver staining, we can see the fine um, processes of neurons. I mean, dendrites, axons, which are arise from the cell body and undergo branching and give very unusual profile to whole cell. I will show you later this type of stainings. Or, for example, in the case of connective tissue, we want to see, for example, identify types of connective tissue. We have many types of connective tissue. And when we are talking about the loose areolar connective tissue, for example, uh, we have the reticular tissue, we have the tissue which, for example, um, loose areolar connective tissue. So reticular tissue has reticular fibers in the extracellular matrix, and they are visible only if you stain it with a silver staining. So I try to show you this. Look, neurons and wait. Look, lymph node, which is stained with the silver staining, and via which we are able to identify their reticular fibers. So this is whole lymph node. These are lymph nodules, these small round-shaped structures here. This is connective tissue capsule, which covers the surface of this lymph node. And if we magnify this image, look, what we see here, what is this? Blood vessel, of course, another group of blood vessels. What is this one? Look, do you remember this tissue? Fat cells. Yes, this is adipose tissue. And if you go to the parenchyma of lymph node, you see these dark stained lines, black stained lines. These are reticular fibers, which are not observable in the case of, for example, hematoxylin eosin staining. Okay? So I want to show you also the cells stained, I mean, nerve cells stained with the silver. I think I should have here this. Look, this is neuron, pyramidal neuron, human pyramidal neuron. This is cell body. Cell body. Do you remember cell bodies of spinal cord, which you see on previous slides? And here you see also the processes of these cells. Look, dendrites here and there. So this is cell body again, this triangularly shaped, and these are processes which are not actually visible in the case of um, hematoxylin or eosin staining. So this is epithelial cell, which is represented here via the light microscope. Look, this is LM image of epithelial cells. What you see here, you see the borders of the cell, you see nucleus, nucleolus, chromatin, look these dark dots, and you see also the cytoplasm very well. And in cytoplasm, you see these granulations. So well stained rough endoplasmic reticulum. This is TEM image of this cell, TEM image. And in look, this is only one cell here, which is fully represented in tissue sample. You see neurons, you see, sorry, nucleus of the epithelial cell of this cell. You see, sorry, nuclear envelope, and in some places by these two leaflets of nuclear envelope are clearly identifiable. Plus, you can see here mitochondria, look, profiles of mitochondria, profiles of lysosomes, endoplasmic reticulum. Look, this is endoplasmic reticulum. But guys, this is small, zoom is quite low. I will show you this image um, with a quite high zooming. In this case, you will see more details. But I want to show you the whole image and to um, compare this with the other images obtained with a different type of microscopical techniques. So light microscopy, you see cell shape, shape of nucleus, you see cytoplasm and some type of granulation, but you are not able to identify organelles in the cell. Transmission electron microscopy, if you use the small magnification, you can see the whole cell. It depends on type of cell. For example, even neurons, pyramidal neurons or Purkinje cells, we are not able to see the whole neuron in the, even if we use small magnification of transmission electron microscopy. But in light microscopy, of course we can. But to observe the fine details, I mean organelles, we have to go to the transmission electron microscopy, Golgi complex. And look, these dark dots in the cytoplasm, they are ribosomes. Transmission electron microscopy is able to obtain the images even of polyribosomes too, but not subunits of ribosomes because they are too small. So cell membrane is clearly visible in the transmission electron microscopic image. And you see also extracellular space between the cells. Look, which is, and there you can see these dark dots or thread-like structures. So these are usually fibers, you know, collagen fibers, different type of collagen fibers. So when the collagen fibers are transversely sectioned, you see their profiles as a dots. If they are longitudinally um, sectioned, you see their longitude thread-like profiles. So this is image of epithelial cells, which is obtained via the um, scanning electron microscopy. And this is image of epithelial cells, which is obtained via the atomic force microscopy. So about these two type of methods, you see that they are three-dimensional. They give three-dimensional views of cells. You see here the bulging nucleus, but the same is here. Look, the cytoplasm, uh, surface view of nucleus, surface view of cytoplasm. You can see also here the even cytoskeleton of cells, different components, microtubules are observable in the cell, and sometimes microfilaments too. But you see here cell membrane. So the resolution power of atomic force microscopy is also very high. Plus, it gives us possibility to obtain even live cells in real time, what is really very important for studies, for research. So we have in our lab, we have this, research lab, we have this atomic force microscopy. And later, of course, you will have possibility to see this microscope too. Okay, so another tissue. Look, this is connective tissue. And connective tissue contains a lot of extracellular matrix and contains cells in the matrix. These are fibroblasts, main principal cells of connective tissue. Uh, Look, fibroblasts are quite active cells, and you see here the fibroblast in TEM images too, with their nucleus, with their cytoplasm. This is small part of cell, of course. You see that the clearly visible is here nucleus with the prevalence of euchromatin. What you see in the cytoplasm is also rough endoplasmic reticulum, and you see also here mitochondria. I want to show you, this is a scanning electron microscopic image of fibroblasts. Look, and these are blood cells, and these are fibers, collagen fibers. Look, this pink stained collagen fibers, and these dots and thread-like structure. They are also collagen fibers, and their shape depends on the plane of section. But here you can clearly see a three-dimensional view of the collagen fibers. The same is, for example, in the case of atomic force microscopy. So how these two microscopical techniques, I mean AFM and scanning electron microscopy, works, we have talked about this next week. But now I want to show you the uh, this is muscles tissue, nerve tissue, And I want to show you the organelles of cells obtained. Oh, look. Um, you see here, um, previous slide here, small part of nucleus. This is one micrograph, this one, and this is another micrograph. Look, I will show you. So 2 micrographs are here. Okay, this is part of the cell which demonstrate the this is quite high magnification, and you see here nucleus, small part of nucleus. Look, this is nerve cell, neuron. Neuron is here too. This is LM image, LM micrograph, and these are TEM micrographs. Look. So look. So we have here profiles of nerve cell, neuron. And you see here nucleus, and small part of the nucleus is represented here in TEM image. And you see the cytoplasm. Whole cell body is clearly visible here. And granulation, I mean, this is toluidine blue staining, that is, or basophilic staining. It means everything what is stained here well contains nucleic acids and sulfate groups. So the small part of nucleus is represented here. You see the 2 leaflets of nuclear envelope. You see the chromatin. This is heterochromatin. This is euchromatin. Plus, you see a small part of cytoplasm, which contains mitochondria. Look, this is mitochondria. Again, 2 leaflets of mitochondrial envelope is clearly visible. I mean, membrane, mitochondrial membranes, and cristae are also identifiable, profiles of cristae. So you see here also polysomes. Look, 3 ribosomes, which give rise to the polysomes. Cell membrane of this cell and cell membrane of other cell. Another small part of cell cytoplasm is represented here, and what you see here, again, small part of nucleus, and you see Golgi complex and rough endoplasmic reticulum. Look, this is rough endoplasmic reticulum with a set of membranes. Look, set of membranes, and also the polysomes are identifiable between the set of membranes and on the surfaces of these membranes. Profiles of mitochondria are also clearly visible here. This is much more higher magnification. That is why you see the, look, this is rough endoplasmic reticulum, this one here too, this one, polysomes free floating in the cytoplasm and also attached to the surface. Look, these dark dots, they are ribosomes which are attached to the surface of rough endoplasmic reticulum. This is Golgi complex. Again, C phase and T phase. small part of nucleus with two leaflets of nuclear membrane and multivesicular body and mitochondria. So what are multivesicular body? About this next time when we start to discuss content of the, I mean, cell organelles. Okay, so okay guys, of course, if you have questions, please write me the questions. And actually, I'm done with my tutorial. Please write me your questions. Um, and I will try to explain, re-explain, or clarify this everything. You can go and visit this histology guide webpage, which is very interesting, very informative. So it was very nice to have a possibility to talk with you and to give you explanations and descriptions of this very important part of histology and cytology. So see you, meet you next week. Bye. So sorry, I'm not. Everything is clear. Thank you. Will you please provide example questions? Of course, I will provide example questions. Two quick question if you passed part of life science one as well. So, now the situation with the life science subjects and with the um, body system is changed. So previously, histology was part of life science. Now it is part of body system. So in life science, you will have um, molecular biology, biochemistry, and cell biology. But in the body system, you will have, beside the anatomy and physiology, you will have histology and you will have also embryology. So, what else? Another question. I will check. Okay, so I think I'm done with your questions too. So, when will we have our exams and how many exams? I think you will have two midterms and you will have one final, of course, but two midterms, no quizzes. Okay, two midterms. And usually, when you will know this data of date of your midterm, and previously 2 or 3 weeks earlier, I usually prepare a set of micrographs which I upload on the Google Drive, and topics, I mean syllabus of midterm, using which you will have possibility to prepare for midterm. What else? Is everything clear? Okay.
Yes, thank you.
Thank you for your attention. And so meet you next week. Bye, guys. You will have now anatomy.
Okay, bye.
Bye, 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 bye.